Everyone, sorry we're not having class today, but we'll do this instead. Starting into chapter 10, looking at genetics. So some of our announcements. If you haven't done so already, you'll need to take the Kahoot quiz. And this is the quiz based on what we covered on Friday. So I posted the link in eCampus, which you probably saw whenever you clicked on this lecture video. Also, as we go through here, there'll be a participation assignment. Since we're not actually in class to do groups, there's still a question that is similar to something we would have done in class. So you just need to write down the answer to those to turn in on Wednesday when you come to class for your participation points for today. And then on Wednesday, we will have a quiz on what we're talking about today. So our learning outcomes for this part, defining heredity, talking about Mendel's 5 hypotheses, how to do Punnett squares. So this is something we looked at last week in lab, Mendel's first and second laws, and then actually kind of recapping gene expression, talking about how genes influence traits. All right, so first of all, Gregor Mendel is the father of genetics. I posted a link also there in eCampus to this video, which is just a nice video to watch about genetics in addition to this. But he talks about genetics and the idea of heredity, which is just passing on traits. So again, heredity is passing traits on to the offspring. And we do that by passing on DNA, and that's what we just finished talking about with meiosis. So the production of gametes is how we pass our genes on to our offspring. So working with mainly pea plants, he came up with 5 hypotheses, 5 things that he noticed while investigating how genes were passed on. So one was that parents don't transmit traits directly to the offspring. So for example, you don't necessarily give kids blue eyes. You don't give them the blue eye, but you give them a gene that then codes for that protein. So they transmit, instead of the actual trait, they transmit the information. Now, you have to understand what he was coming up with. This was before they understood DNA and the structure of DNA and how it was passed on. He was just observing how things seem to be passed on. Now we can go back and kind of fill in that information. So again, now we know that genes are how we transmit those traits onto the offspring. He also came up with the hypothesis that each parent has 2 copies. He called them factors. Again, now we call them genes that govern the traits. So since there are 2 copies, those 2 copies are either the same, which is homozygous, or they're slightly different, which is heterozygous. So another term, allele, is an alternative form or variation of a gene. So again, in lab, we looked at some of those as well. But again, eye color is a good example. It's the one I used. So if somebody has blue eyes or green eyes or blue eyes, it's because they have slight variations of a gene that makes pigment in the eye. So they're not completely different genes. They're just like variations, and we call those alleles. So if those alleles are the same from each parent, it's homozygous. If they're different, it's heterozygous. All right, then again, these alternative forms of the factors are referred to as alleles, and that is what leads to the variation. And so those alternative traits, though, that appearance is determined by which alleles actually are received. So again, a couple terms that were covered in lab last week. So genotype, those are the actual genes. So which alleles were received? Are they homozygous? Are they heterozygous? We'll talk here in a minute about dominant and recessive. So again, which genes were actually receive, that makes up the genotype, then what those physical characteristics turn out to be, again, eye color, skin tone, hair color, height, all that kind of stuff, the physical characteristics, that would be the phenotype. And that would be based on what we're looking at here in this chapter. Are they dominant? Are they recessive? How do they get expressed? So back to gene expression. And then the two alleles of an individual do not affect each other. Okay, now we'll go through Mendelian genetics first, and then non-Mendelian. So this one didn't turn out to be completely accurate for all traits. But again, the idea that the two alleles don't affect each other was one of his hypotheses. And then our last one here, so the presence of an allele does not ensure that the trait would actually be expressed. And this is what we see. So if a trait is dominant and another then is recessive, so if one allele is dominant, it means that even if somebody is heterozygous and they have one dominant allele and one recessive allele, that dominant allele will mask the recessive one. So even though I may have an allele for blue eyes that's recessive, if I also have a brown eye allele, that dominates over the blue, so I end up with brown eyes instead of blue eyes. All right, this figure here is just showing some different traits and whether they're recessive or dominant. So a couple of the recessive traits to mention here. So baldness. So if the only way that an individual becomes bald is if both parents would give them that recessive trait. And that leads to that hairline receding with age and ultimately into baldness. But again, as long as an individual has one allele that doesn't cause baldness, they would not become bald, only if both parents give them that recessive trait. Also, being albino, which is a lack of melanin pigmentation, that is caused, again, recessive. 2 copies of that would have to be received. Red-green colorblind is another example of a condition or substance where, again, both traits would have to be the recessive trait. A couple examples of the dominant traits, so again, having just one allele in these conditions would cause this trait to be seen. So for example, mid-digit hair, individuals that have hair on that mid-digit, they only need one of those alleles, so they could receive a recessive and the mid-digit hair trait, and they would still express that trait because, again, it's dominant. And then brachydactyl, which is short fingers. Again, that is also a dominant trait. So just having one allele would cause that phenotype to be expressed. All right, so here we have a Punnett square. And as we talked about in lab last week, this is a way to visualize the possible offspring whenever we know 2 parents' genotypes. So if we kind of look at this, remember males and females, of course, we are diploid. But whenever we go through meiosis, our gametes are haploid. They have one of each chromosome. All right, so here in this example, we're going to use, we have 2 heterozygous individuals that are mating. So we're going to say here on the left-hand side, this is the female. And again, she was heterozygous, so her parents, one gave the dominant and one gave the recessive allele. So when her cells go through meiosis, half of the eggs would have the dominant trait and half of them would have the recessive trait. So if these are her eggs, these are the possible traits for this gene in her eggs. She's either dominant or recessive. Same thing for the male. If he's heterozygous from his parents, when he goes through meiosis, half of his sperm will have the dominant trait, and then half of those sperm would have the recessive trait because he's heterozygous. Now, what these Punnett squares show are, again, likely the possible combinations that can result as long as we know the genotype of the parents. And it doesn't matter whether you put the male on the left and the female at the top, or vice versa, as long as the male's on one side and the other, as long as both of them are represented. So we have eggs and sperm being shown. Now, I will mention they use P for this, I think because of Punnett square. P isn't a great letter to use for traits because it can be hard to tell the difference between capital and lowercase letters. So like A, T, R, those are better letters because it's easy to tell the difference between a capital letter and a lowercase. But we'll go with what they have here. Now below it's just showing how you would create this. So if you know the male and female genotypes, again in this case we're going to put the eggs, so the female genotype here on the left, and then we're going to put the sperm at the top. But we can just as easily switch those. All right, so what the Punnett square does is it shows, again, fertilization is random. So which egg and which sperm actually come together during fertilization is completely random. So in this first square, in this case, the egg is giving a recessive trait and the sperm is giving a recessive trait. So one possible outcome for this zygote or this child would be homozygous recessive. Okay, then we could also have the recessive trait from the egg pairing with a sperm that has the dominant allele. So if those two come together, then the child would excuse me, be heterozygous. Right, so then our next possibility, here we have the egg with the dominant trait, the sperm with the recessive, so we get another heterozygous. And then finally, we have the egg giving the dominant trait and the sperm also having the dominant trait. So if these end up coming together in fertilization, we get the homozygous dominant. So when we put all that together, our Punnett square from this cross, whenever we have two individuals that are heterozygous, now we can look at the percentages or the ratios of the offspring. So some possible genotypes, there's a 25% chance or 25% chance that a child from this mating would result in being homozygous dominant. Now we can show this as either a percentage or you can also show it as a ratio. So there's 1 out of 4 chances of this, so 1 to 4 ratio. There's also a 50% chance, or 1 out of 2, that they would be heterozygous. So there's one here and here. So we have 2 out of 4 chances of being heterozygous. We just reduce that to 1 out of 2. And then finally, there's also a 25% chance that the baby would be homozygous recessive. Okay, now if we look at the phenotype from this, which is the actual physical characteristics of what this child would look like or display, now we have to come back and think about dominant and recessive. So we'll use the example of dimples. So having dimples is a dominant trait. So you only need one allele that codes for dimples, and you'll express that. And so if P here is representing dimples, the possible phenotypes, though, there's a 75% chance that they would have dimples. Okay, so if I kind of clear all this here so we can see. All right, so now again, there's a 75% chance because the homozygous dominant would produce the dimples, but also the heterozygous offspring would also show dimples because it is a dominant trait. As long as you have one dominant allele, they show that characteristic. So there are 3 out of 4, 75% chance of having dimples. There's only a 25% chance that the child would not have dimples. So again, these Punnett squares, we can set these up, and that's what you'll be doing for your participation is doing some examples of these. All right, and this is just showing another example here in Snapdragons, looking at another trait. So again, they're still using P, in this case for purple, because purple color is dominant to white. So in order for this flower to be white, it's recessive, so they would have to be homozygous recessive to see this trait. So what this is showing is if we have a cross where we have a homozygous recessive crossing to a homozygous dominant, what you can see, so we call these a true breed or a purebred. So if they're homozygous dominant and we're crossing to a homozygous recessive, we call this an F1 generation, where the parents were both homozygous, one dominant, one recessive. We see that the phenotypes, it's 100%, they are all going to be heterozygous. So the genotype is 100% heterozygous, and it's also 100% purple. So the phenotype of all of these offspring are going to be purple. So if you made this cross, again, all of those would end up being purple. Now, where it gets kind of interesting, though, is if you would take these heterozygous children and cross them, now what we see is similar to the example we just did. So now if both of them are heterozygous, so they look purple, okay, but they're purple with a heterozygous genotype. So again, if you cross two purple flowers, you wouldn't know, without doing like gene sequencing, you wouldn't know whether that purple flower was homozygous dominant or whether it was heterozygous. Well, this is one way you can figure that out. If you would take two purple flowers and you would keep crossing them, if you always got purple flowers, that would indicate that both of those parents were homozygous dominant. But if you started to see some white ones popping into the population, you would know that those purple flowers you started with were actually both heterozygous. Because that's the only way that you could cross two purple flowers and end up with a white flower, is if both of them are heterozygous. So that's what Mendel was doing. He was making all these crosses, and he used all this to come up with those hypotheses. All right, so for your participation assignment, so again, with pea plants, there's a gene that determines height. This is not the case in humans, but in plants, there's one gene that determines whether the plant is tall or short. And the tall allele is dominant to the short allele. So showing, whenever you're writing these out on a piece of paper, draw out your Punnett squares. Determine what the gamete ratio is. So again, the offsprings that are made here. So again, genotype and phenotype. If we crossed a homozygous dominant pea plant with a homozygous recessive pea plant. So set those crosses up. So again, what would that Punnett square look like? So if we have a homozygous dominant and a homozygous recessive pea plant, again, I suggest using T for tall, and it's a good letter to use. If you make that cross, what are the possible ratios or percentages of genotype and phenotype of those offspring? And then what you're going to do is take one heterozygous pea plant and one homozygous recessive. So you're going to set up two different Punnett squares. So you'll have different combinations. So show these two crosses and the percentages. Again, you can either use a percent or ratio of the genotype. So how many are homozygous dominant? How many are heterozygous? How many are homozygous recessive? And then for phenotype, you're just looking at what's the percent of being tall versus the percent of being short. All right, so write those out and turn those in on Wednesday. All right, so just a couple more things for today. So Mendel had a couple laws. So he had those hypotheses, but then he also came up with two main laws of inheritance. The first one, and we've actually already covered this under meiosis because we know this already at this point, but he didn't. So his first law was called the law of segregation, and this is where he was able to determine that those traits that were passed down, somehow they were separated whenever the gametes were formed. Again, we know that they go through meiosis. That's what we just covered. Okay, so we start out with diploid cells, but then through meiosis, we end up with haploid gametes. So that happens with both male and females. So their gametes are going to be haploid. So they segregate or separate those homologous chromosomes. And that's what meiosis does. It divides those so we have one of each for haploid instead of diploid. So that was referred to as his law of segregation. We had to separate those homologous chromosomes during gamete formation. And then the second law we talked about with meiosis as well, which is independent assortment. So this is one of the things that leads to genetic variability. So the way that the chromosomes line up during metaphase is random. So the separation of one homologous pair is completely independent of the segregation of other homologous pairs. So again, the way that our 23 pairs of homologous chromosomes line up in metaphase is completely random. So whether we end up with mom's chromosome 1 and dad's chromosome 5 or whatever combination, that is independent of each other. It's not like because we got mom's chromosome 1, we're also going to get mom's chromosome 2. Each one is completely separate and independent. All right, so then just the last thing, and this is actually kind of a recap, but just trying to put some of this past information together for us. This is showing how the genotype determines the phenotype, which is also what we talked about with gene expression. All right, so if we start here with our genome, which is all the DNA inside of our nucleus, so it's all the chromosomes. Remember, each chromosome has one long DNA molecule, but those molecules get separated into genes. So a gene is a segment of DNA that makes a protein. So on that DNA, if we look here at this hemoglobin gene, there's a gene that would make the protein hemoglobin. So it's a piece of DNA. We go through transcription. So we use that DNA to make RNA. And we go through mRNA processing, and that mRNA leaves the nucleus and goes out to the ribosome, and that mRNA is then used to go through translation to make a polypeptide or a protein. So it puts those amino acids together. Now we have our protein. That protein is going to fold into its three-dimensional shape. So quick recap of proteins. This would be the primary structure, the amino acid sequence. This would be our tertiary, three-dimensional shape. Okay, then that protein then goes and does a job. In this case, it is taking oxygen in from our lungs and delivering it throughout the body through the blood. So again, the gene, if there's a mutation like sickle cell anemia and that gene gets messed up, now our amino acids get messed up, and that can affect the protein, which is what happens with sickle cell anemia. So again, that gene expression is how our genes determine our traits, how genotype determines phenotype. All right, so again, just ending here. So again, heredity is the passing on of traits. Those 5 hypotheses we went over, know how to create and understand, interpret a Punnett square, the first and second laws of Mendel, and then as we just mentioned, how genes influence our traits. All right, thank you guys, and we'll see you on Wednesday.